Hello everyone and welcome to Annotated Medical Tutorials. This is a channel specifically designed to simplify medical concepts. Today we want to take a quick look at the structure of DNA before we go for topics like DNA replication, protein synthesis, and other aspects of genetics. So DNA contains the instructions that our bodies need in life, and it is the one that makes us different from animals and also different from one another. So DNA will produce functional copies which are called RNA, and from these RNA, that's where proteins are going to be made, and these proteins are going to be needed by the body. So the proteins can either be structural or they can be functional. Right, again, the proteins can be structural or they can be functional. So the structure will be unique and it will differ from one organism to the other depending on the DNA, and also the function can be unique depending on the DNA. So right now we need to define what exactly DNA is. So DNA is a polymer of nucleotides, or it's a polynucleotide. This is because let's say a polymer is a structure in which many units called monomers are joined together to form the polymer. So in our case, the monomer would be a nucleotide, and then the structure that will form is DNA, which is a polynucleotide. So these circles are resembling monomers. So the monomers are going to be joined together to form a polymer. So this one single unit is a monomer. But in the case of DNA, the monomer is a nucleotide. So they are joined together to form a polynucleotide, which is the true definition of DNA. For us to understand the detailed structure of DNA, we need to go back to its basic unit, which is the nucleotide. So let's now look at what exactly is a nucleotide. So for us to make a nucleotide, we need three raw materials. And the first raw material is a nitrogenous base. We need a nitrogenous base. These are nitrogen containing carbon rings. And these nitrogenous bases, we've got two types. The first one is double ringed, and then the second one is single ringed. Again, the double ringed ones are the larger ones, they're called purines, and the single ringed ones are called pyrimidines. So the purines are the ones which are bigger, they've got double rings, and the pyrimidines are the smaller ones, they've got single rings. Double ringed ones, we have got adenine and guanine. And for the pyrimidines, we have got thymine and cytosine. Right, listen again. For us to make a nucleotide, we said we need three raw materials. And the first raw material are nitrogenous bases, which are nitrogen containing carbon rings. And we've got two types of nitrogenous bases. The first one are double rings, and the second ones are single ringed. The double ringed ones are called purines, and the single ringed ones are called pyrimidines. The purines are double ringed structures, and pyrimidines are single ringed structures. In examples of purines, we've got adenine and guanine. And for pyrimidines, we've got thymine and cytosine. Now going on to the second raw material we need for a nucleotide, we've got pentose sugars, or simply called pentoses. So they are called pentoses because each sugar has got 5 carbon atoms. So if we are to draw it like this, we've got 1 carbon, 2, 3, 4. There we've got oxygen, 1, 2, 3, 4. And then the fifth one will be here. Now I want you to take note of this fact. If on carbon number 2 we've got an hydroxyl group and a hydroxyl group on number 3, this pentose sugar is called a ribose, and it is used to make ribonucleic acid, which is RNA. So if we've got a hydroxyl group on number 2, it's called a ribose. But if you knock out this oxygen on carbon number 2 and we are only left with the hydrogen, the name changes. Because we've knocked out oxygen, we now call it a deoxyribose. And a deoxyribose is the pentose sugar that will make DNA. A ribose is the one that makes RNA, but a deoxyribose is the one that is used to make DNA. So we are now going on to the third raw material we need to complete the structure of a nucleotide. The third one is a phosphate. So now let's draw the complete nucleotide. Here we've got a 5 carbon sugar, which is a pentose, specifically deoxyribose. And here we've got a nitrogenous base, which could be a purine or a pyrimidine. So the third raw material, which is the phosphate, should be here. And we're going to indicate it in red like this. So these three make a complete nucleotide. So during the process of formation of a nucleotide, there's a structure that is formed first, which contains only the nitrogenous base and the pentose sugar. So when these two are joined, the nitrogenous base and the pentose sugar, the structure is called a nucleoside. Again, a nucleoside is a structure which contains a nitrogenous base and the pentose sugar without the phosphate. And each nucleoside has got a different name depending on the nitrogenous base that's found there. So actually the name of the nucleoside is derived from the nitrogenous base. So let's go on to name the first nucleoside we have. And this one has got adenine as the nitrogenous base. And the nucleoside is called adenosine. I want you to take note of the difference between the names. A is for adenine. That's the nitrogenous base. But the nucleoside is called adenosine. If you've got guanine, it's called guanosine. If it contains thymidine as the nitrogenous base, it's called thymidine, which is thymidine. If it contains cytosine, we call it cytidine. Again, let's look at the definition of a nucleoside. A nucleoside is a combination of a nitrogenous base and a pentose sugar without a phosphate head. So the name of the nucleoside is derived from the nitrogenous base it contains. If it contains adenine, we call it adenosine. If it contains guanine, we call it guanosine. If it contains thymine, we call it what? Thymidine, yes. If it contains cytosine, we call it what? Cytidine. Excellent. Now we want to add the phosphate head. We call it the phosphate head because of the position at which the phosphate is attached. Remember, we've got the 3' end and the 5' end. So the phosphate is going to be attached at the 5' end. That's why we call it a phosphate head. So if it contains one phosphate, it's called a monophosphate. Let's now go into the specific naming of the nucleotide. The name depends on the nucleoside. So if it's adenosine, we call it adenosine monophosphate. If it's guanosine, we call it guanosine monophosphate. It's a monophosphate because there is one phosphate head. If it's thymidine, we will call it thymidine monophosphate. And last but not least, if it's cytidine, we call it cytidine monophosphate. This is simple, right? If we add on another phosphate like this, the name will change. Remember, I said the name will depend on the number of phosphate heads that are there. From monophosphate, we change it to diphosphate. So in other words, if it's adenosine monophosphate, it will be called adenosine diphosphate. Then guanosine diphosphate and thymidine diphosphate, cytidine diphosphate. Again, let's go on to add the third phosphate head. So like this. Now that we've got three phosphates, we change the name to triphosphate. From adenosine diphosphate, we go to adenosine triphosphate, guanosine triphosphate, thymidine triphosphate, and cytidine triphosphate. So adenosine triphosphate is shortened as ATP, guanosine triphosphate shortened as GTP, and thymidine triphosphate is shortened as TTP, and cytidine triphosphate is shortened as CTP. As we all know that ATP is the major energy currents inside the human body. This sounds familiar, and it has got 1, 2, 3 phosphates, adenosine triphosphate. Again, as we have said that we have got carbon number 1 here, number 2, number 3, number 4, and number 5, which contains the phosphate head. This information is important in determining the direction of DNA because it runs from number 3 to number 5. So we call it polarization. So it runs from 3' end to 5' end. We will later use this information. Now let us get into the process of joining these nucleotides. And I want to emphasize the fact why we were adding more phosphates until we got to a triphosphate. So remember, we've got a nucleotide here which runs from 3' to 5' end. And then we've got another nucleotide here that we want to join with the first one. So the phosphate head of the nucleotide here will go to join the hydroxyl group on the third carbon of the first nucleotide. But if it goes like a monophosphate, it will be rejected because it doesn't have enough energy for the reaction to take place. So it won't bond. If we add the second phosphate as a diphosphate, it will still be rejected because it does not have enough energy. But if we use a triphosphate now, it has got enough energy for that bond to form, and it will be joined, and a phosphodiester bond is formed, which is a very strong bond. Let me repeat again so that we get this concept. For us to join two nucleotides in the polymerization or formation of DNA, a monophosphate cannot form the phosphodiester bonds needed, neither does a diphosphate form, but a triphosphate, only a triphosphate has enough energy to form the phosphodiester bond that's needed in the polymerization of DNA. So now let's draw the joined nucleotides in a DNA polymer as a strand. So here are the pentose sugars, and there is the phosphodiester bond, and there are the nitrogenous bases. This bond that's formed is called the phosphodiester bond, and it is a very strong bond which cannot be broken by simple temperature changes or simple pH changes. It will only take some enzymes, very strong enzymes, to break these phosphodiester bonds. Like we've said before, that this side we've got the 3' end, and that other side we've got the 5' end. This is what we call DNA polarization. Another characteristic of DNA to note is that it doesn't exist as a single stranded structure, but it exists as a double stranded molecule. This is achieved by a complementary base pairing. So these bases, they will be running alongside each other like that. So what we call complementary base pairing is that adenine pairs with thymine, cytosine pairs with guanine, like that. Please note that the other strand of DNA is also polarized, but it runs from 5' end to 3' end. So these are anti-parallel now. The complementary base pairing is via the hydrogen bonds. So adenine pairs with thymine via 2 hydrogen bonds, but guanine and cytosine pairs via 3 hydrogen bonds. And this is of paramount importance to note. Again, nucleotides in a DNA strand are joined together by phosphodiester bonds, which are very strong and they cannot be altered by any temperature changes or pH changes. But between the two strands of DNA, hydrogen bonds, which are weak, the bonds which join the nitrogenous bases via what we call complementary base pairing. And these hydrogen bonds are weak, that they can be changed by temperature changes and also pH changes. And also we said the first strand runs from 3' to 5', and then the other one runs from 5' to 3'. And we call this anti-parallel. So when we are describing them, we say the two strands are anti-parallel to each other. So for each strand of DNA to be made, there are some enzymes that are needed. So for the formation of one DNA strand, nucleotides are joined to form polynucleotide, and the enzyme responsible for that phosphodiester bond is called polymerase, because they are forming a DNA polymer, so we call them polymerases. So formation of the diester bonds is done by DNA polymerases, but breakdown is done by a different enzyme which we call phosphodiesterases. So because the phosphodiesterases break down nucleic acid, we call them nucleases. And there are different types of nucleases that we need to talk about depending on the position at which they cut the DNA. So here is our single DNA strand, which is polarized. We've got 5' and 3' end. At the 3' end, we've got a free hydroxyl group, and then on the 5' we've got a free phosphate group, like that. So now let's talk about the enzymes that break down DNA. Some can go to the extent of breaking it down from the ends. Some can break it down from the center. So remember, since we said they break down the nucleic acid, we call them nucleases. But some are breaking down DNA only, and we call them DNases. DNases. Some break down RNA, and we call them RNases. Right. So the ones that break down DNA from the ends going inwards, we call them exonucleases because they break down starting from the ends. Those which break down DNA starting from the center are called endonucleases. Endonucleases break down it from the center. Remember, we said that DNA exists as a double-stranded structure via complementary base pairing, and it is polarized, and these two strands are antiparallel to each other. But I want you to take note of the fact that it does not exist as a simple structure like this, but it will then coil to form a double helix. And this will hide away the weak hydrogen bonds from the environment. Otherwise, they could be changed and DNA structure could be tampered with. So the DNA double helix has got grooves which are divided into minor and major grooves. For example, this one is a minor and that one is a major groove. That one is a major groove. This one is a major groove and this one is a minor groove, major groove. So these grooves are very important in the fact that some drugs can bind to those grooves and occupy them depending on the type of drug. So some grooves can occupy the minor, some drugs can occupy the minor groove, and some can occupy the major groove depending on the type of drug. So this will influence the activities of DNA, including DNA replication and even protein synthesis. So let me give you an example of a drug that occupies the minor groove. So when it occupies the minor groove, it stops DNA replication. And this drug in particular is called dactinomycin. So when dactinomycin binds to the minor groove of DNA, it will stop DNA replication. In that when it binds to the minor groove, it will stop even the unwinding of DNA so that it cannot be replicated or copied. So this is of paramount importance in the treatment of cancer, in that it can be used as an anti-cancer drug. So dactinomycin is used in the medical industry as an anti-cancer drug. So we have come to the end of this lecture where we're looking at the DNA structure. Now that we are done, we can be able to move on to the next topics, which include DNA replication and protein synthesis and other topics that are found in medical genetics. If you haven't yet subscribed to this channel, please click the subscribe button and the notification bell. Thank you.